I am Nurul Nazirin. I am a protein structural analyst and programmer with PDBe. I will be presenting today's webinar entitled Creating Complex PDBe API Queries. And if you look at the current slide, there is a link there to our API page. Just go to pdbe.org/API. This is the third webinar in a series of 6. There were 2 previous webinars on General Introduction and PDBe Search API. There will be 3 more upcoming webinars ending on October 20th. To register for future webinars, please go to the link that I am pointing out here, PDBe Europe GitHub link, or alternatively, you can go to pdbe.org/events. You would have to register for each webinar individually. We will upload all webinar videos, slides, and Jupyter notebooks to the same URL. A summary of what this webinar entails. First, I will talk a little bit about what type of data is in the PDB. And then I will briefly introduce you to the 3 types of APIs that we provide. And then I will narrow it down to what we are focusing on today, which is the entry-based API. And I will then explain the categories of endpoints within the entry-based API. And I will relate to how we at PDBe use these endpoints. And finally, I will end this webinar with 2 use cases, which uses multiple endpoints to showcase how to use our REST API. So what type of data is in the PDB? The PDB contains 3-dimensional structures, data of biological macromolecules consisting of proteins, DNA, RNA, and bound ligands. We here are at PDBe in the UK, together with RCSB, PDBJ, and BMRB. We make up the worldwide PDB, or Protein Data Bank. EMDB will soon join WWPDB, and the data archives are made up of EMDB, PDB, and BMRB. We have a deposition system called OneDAP, which is shared across all the sites. What OneDAP allows is to allow depositors to add data, either during deposition or the data are added during curation. And the kinds of data that are added are experimental methods, sequence informations, connectivity, for example. And these data are added to the archive MMC file or validation XML file. We also have enriched data by mapping to external databases such as PFAM, Interpro, and CAF. This is made possible based on the mapping between the sequence database UniProt to our structural database PDBe. And this is called CIFS. CIFS is a collaboration between UniProt and PDBe. We offer 3 types of APIs. If you go to this link, pdbe.org/API, this is what you will see. This is the landing page. And you will get 3 different links to 3 different APIs. The one that is highlighted here is what we will talk about mainly today. This slide briefly describes the 3 API. The first API is a Solr-based query API. To use this API, you would have to be proficient in Solr, and this has been the topic of last week's webinar given by my colleague John. Then the second API is aggregated API. It has Neo4j graph database at the back end, and this will be the topic of next week's webinar given by a fellow colleague Srinath. And entry-based API is what we will be talking about today. The back end is based on an Oracle relational database. Both the entry-based API and aggregated API need an input which is entry specific. That could be PDB ID or chemical component ID or UniProt ID, for example. Whereas as mentioned, querying by Solr query system, you need to use data specific to Solr. One thing that you can do, and what a lot of people do, is to use the query system to obtain a data set of PDB IDs. And this data set is then used as input for both the entry-based API and the aggregated API. It is worth mentioning that the entry-based API is used to populate data on the PDBe entry pages, whereas the aggregated API is used to populate data on PDBe KB aggregated views. To view the presentation given by my colleague John for Solr-based query system, you can use this link that is highlighted here. It is the same link given earlier. There are a set of tutorials and as well as the video of the webinar. And just to emphasize, this is what we will be talking about today, which is the entry-based API. Now, let's take a look at the entry-based API. There are 8 categories within this API, and in the UI, they are each represented by a tab. There is also a tab for the search API, which is actually the query system, the Solr-based query system. This is how the documentation looks like. Let us take a look at the first tab, which is the PDB tab. Each tab has their own URL, as you can see here. For example, this is the URL for the PDB tab. Each of these that I'm highlighting are endpoints. For example, there's an endpoint called summary and another endpoint called molecules. These endpoints here have been collapsed, whereas this particular endpoint called Ligand has been expanded. Whenever an endpoint is expanded, this is the view that one would get. On the left-hand side is a list of endpoints. Let's take a look at how the documentation page works. Right underneath the endpoint list, you will find that there is a URL that you can use to go to each endpoint. Following that is a short description of the kind of data that each endpoint provides. Following that, you will get or you will see a form. The first field is for you to try out get post. For example, here, the user has input 1CBS, which is a PDB ID. And the second form or the second field in this form is to try out post request. Let's try out the get request. After inputting 1CBS, what the user would do is to click this button here. It's called run call. Once that button is clicked, a new view appears. What this view shows is, first of all, it shows whether you have made a GET request or a POST request, and it provides the actual GET request URL, provided that you made a GET request. What you can do is you can copy this URL and put it in your browser if you want to look at the results in a different window. Followed by that, it details the HTTP status of your request. In this example, it's returning a 200 HTTP status because it is a valid return. Following that is the actual output. And this output is in JSON format, which means that they are in key and value pairs. And if you would like to know what each field means, you only have to hover over them. For example, if you hover over this field called preferred within the assemblies key, it shows what this means. It means that this indicates if the assembly is the preferred assembly or not. And as I mentioned earlier, the second part of this form is to try out post data, and it's very easy to do. What you have to do is provide multiple PDB IDs, for example, for this call called summary. And these IDs are separated by a comma. Simply do the same thing, click on run call, And now it shows that you are making a post call without any ID here. It only shows the endpoint. And here it shows the same formatted JSON as output. Our entry page is powered by the API itself. For example, let's take a look. We are still on the tab called PDBe here. This is our entry page. This section here is powered by an endpoint called summary within the PDBe tab. These two sections here are powered by the publication endpoint. There are two endpoints called ligands and carbohydrate polymer that are responsible to populate data on ligands and environments section. The structure analysis has assembly data, which depends on the endpoint called assembly. There are also details on experiments and validation. Validation actually depends on a different tab called validation. However, the experiments bit depends on an endpoint called experiment within the PDBe tab. It's worth mentioning that we have an endpoint called related publications. This endpoint provides the data that you see if you go to our page, which details mention without citation. Mention without citation is unique to us. Another very useful endpoint is called files. If you go to our page and you click on download, there will be a drop-down. This drop-down lists all the files that you can download. The files endpoint gives you directly all the URLs of a specific entry. Therefore, you can programmatically download all of them. This is an example of how a typical JSON output looks like. There will always be an ID for any endpoints. And this ID is either PDBe ID or chemical component ID. Typically, the JSON for our API is in the form of a dictionary of, a list of dictionaries. In this endpoint, which is an endpoint called molecules, each dictionary is a dictionary of entity information. And there are 3 dictionaries here. However, this one is collapsed. This is one entity, the second entity, and the third entity. Molecules call provides entity level information. And as you can see, this entity here, entity 1, is a polypeptide, whereas entity 3 is a bound ligand. So the molecules call provide data not only on proteins, but also on the ligands. Not all of our endpoints are in the form of the list of dictionaries. However, the majority of them are. Let's move on to the compounds tab. So the compounds tab contain endpoints related to small molecules or PDBe chemical components dictionary. A very useful endpoint is this one. It's called PDBe entries containing the compound. What you will do is you will have a chemical component ID of your interest, input that into the form in this documentation, and what you will get is a list of PDBe IDs containing the chemical component of your interest. This is a good starting point to build a dataset that you want to study your compound of interest. This is the summary call for compounds. And in this call, you will get the basic information for the component of your interest. For example, here is ATP. It has the SMILES information, in keys, for example, and the formula. Another endpoint that is useful is the compound mapping endpoints. It maps your compound of interest to DRUGBank, for example, EC, Kebi, and Campbell, among others. And we use this endpoint to populate data in our pages as well to give links to Kebi and Campbell. Moving on, the EMDB tab provides information on EMDB data. The SIFs tab provides information on mappings to external databases, for example, UniProt, Interpro, and PFAM. And SIFs is used widely. However, one of the pages that uses SIFs almost in entirety is the function and biology page at PDBe. For example, this mapping here is what populates this bit of the page. Another very useful call, if you are interested in mapping between sequence and structure, is the best structure call. This call returns the best PDB structures mapped to a certain UniProt ID. So the input is a UniProt ID. And the list of PDB structures are sorted by coverage. The no-click mappings tab lists down 3 calls relating to entries mapped to RFEM. The PISA tab lists entry points related to PISA surveys. The validation tab is widely used as well. These are actually an exhaustive list of all the endpoints in the validation tab. And the validation endpoints are used to populate data on our experiments and validation page. For example, this image here was generated based on the data given by these endpoints. This is Molstar, our 3D viewer. There is a choice to turn on validation report, and if you choose to view your structure based on the constraints of validation reports, Molstar actually consumes these API endpoints to populate those values. And finally, the last tab is called topology. There are only 2 endpoints for this, and it is used to generate a topology diagram. Let's take a look at the details of entry-based API. Both HTTP GET and HTTP POST requests are allowed, GET for individual end queries and POST for batch queries. Not every call allows POST requests, however, the majority do. The output is JSON in JSON format. There are 3 possible HTTP status codes. The first one is 200, which means everything is okay. The second one is 404. That doesn't mean everything is not okay. It simply means that what you are looking for is not present. For example, in the entry ID 4J1, it does not contain any SIFs mapping. So if you ask for SIFs mapping, you would get 404. The third one is if you get 500 error, it means that something bad has happened, and please report to us if this occurs. We do not have any fixed release cycle. What typically happens is that if we have a new call or new fields, that's when we make a release. We keep backwards compatibility in terms of field and value names since the very first version. We use most of these endpoints ourselves in our pages. So what that means is that the majority of calls are subjected to weekly testing. We have a mailing list called PDBe API users. And if you would like to join, simply send an email to this link. And it's very useful to join this mailing list because whenever we have a release, we send out emails to the mailing list prior to the release so that you can test new features on www.dev. And this usually happens months to weeks before any release happens. I will now guide you through 2 case studies. These case studies attempt to use multiple API endpoints to showcase the relationship between different endpoints. The first case study is trying to compile a data set to study ligand binding domains. And the second case study will be on validation. Let's take a look at case study number 1. We have 4 objectives in this case study. The first is to find PDB entries containing a specific ligand, and we have chosen RHEA or retinoic acid for this. Once we have those PDB entries, we will try to analyze the domain composition using a different endpoint, which is the CIFS endpoint. Using the same CIFS endpoint, we will try to find cath domains which interact with RHEA. And finally, we will reconcile the 2 endpoints to find a set of chains in the entries that contain first cath domains and second cath domains that bind to RHEA. The first endpoint that we will take a look at is in the compounds tab. It's called in PDBe. What you would have to do is to search for retinoic acid, which has the chemical component ID REA in the form, and simply click run call, and you will receive a return of 31 PDB IDs. Now, we have 31 PDB IDs. Let's take a look at the domain composition of the entries. We will take a look at the second endpoint within the CIFS tab, and it is the first call in the CIFS tab called mappings. Because we have multiple PDB IDs, there are 2 choices for what you can do. You can programmatically request this link, which is the CIFS endpoint, for each PDB ID, and that means that you make multiple GET requests, or you can make a single POST request using the entire PDB ID list. This is how the output looks like. The output for CIFS mapping call is the mapping to external databases, for example, here cath, a mapping to PFAM, and a mapping to Interpro. Now let's do a simple data analysis. Using this JSON output, what we can do is simply count the type of mappings. For example, how many entries map to PFAM, how many entries map to cath. Here is showing only for 1 entry, which is 1 CBR. And what you have to do is you have to iterate through the JSON output, which is this, and the PDB ID. For example, here is 1 CBR, and the domain type, which is the field immediately following the PDB ID. And if you go through that exercise, you will find that all 31 entries have PFAM mappings, Interpro mappings, Uniprot mappings, and Go mappings. Let us focus on just 1 mappings, and let's just choose cath mappings. So we know that 26 entries have cath mappings, but how many cath superfamilies are within those cath mappings? Let's count that. So to do that, what we have to do is simply first to look through the JSON output again. And if the domain type does not contain cath, we continue. Otherwise, we take a look at one specific field called homology, which is specified here. So a lot of exercises in REST API consist of going through the JSON output and delving deeper into the fields within those JSON output. And once we store the homology value, what we get is a profile of how many entries contain what domain and what is the homology name of this domain. Next, let's bring chain ID into it. So this is the same JSON output. However, now we are interested in one particular field called chain ID. Now we have moved on from homology, and we will move on to mappings because the chain ID is within the mappings field. So again, you have to go through the JSON output and get the variable from within the JSON output. And let's store this as DOM info chain ID. So once you store that, again, programmatically, you can go through the entries, and this is what you will get. A map of chain ID for each PDB entry that maps to the domains. Now, let us look at another call now called binding sites. This call is within the PDB tab. We use the same dataset that we obtained earlier, and we repeat the steps that we had earlier. However, now we're using a different endpoint called binding sites. And this is the output that you get for querying binding sites. So what we want to do now is to find binding sites for RIA. And that is actually done by getting the ligand residues that contains RIA. And again, we do the same exercise that we did before. However, now we focus on binding sites endpoint. And then again, we search for chain ID and map the chain ID to the ligand residues field for this particular call. Once we have done that, what we can do is cross-reference what is in the binding site chain ID, here referred to as residue info chain ID, with the domain info chain ID, which is what we obtained based on the compounds and SIFs call earlier. Once you do that, you have used multiple API endpoints to generate a dataset, and this dataset we can use for further analysis. So we have this dataset of chain IDs, and we know that this chain ID binds to RIA, and that these chain IDs contain cat domain. In this particular example, they all contain cat domain lipocalin. Let's move on to a second case study. In validation, sometimes we get an unexpected feature. So an unexpected feature is not always a modeling error. What we can do is to generate what is called the quality profile. It is an exercise in looking at multiple copies of a PDB entry, for example, and finding out, for example, if there are 5 copies, 4 copies have one Ramachandran feature, and another 1 copy has a different Ramachandran feature, we could then ask that 1 copy, is something wrong with that 1 copy? In this case study, let's generate a quality profile of multiple copies of a bacterial chaperonin called Groel in PDB entry 1kp8. So the first step that we do is for 1kp8, we look at how many copies there are of Groel. And to do this, go to the PDB tab and access the molecules endpoint. And again, all you have to do is input 1kp8 into the form and click run call and look at the JSON output. So again, what you can do is, because this field here is quite limited in size, so you can copy the URL here and paste it into your favorite browser. And let us take a look at the JSON output. So the molecules call is based on entity information, and what we are interested in is a chaperonin. The first thing we can do is we can filter out based on molecule type. There's a field here called molecule type. This is, for example, entity number 1. This is entity number 2. Entity number 1 is a polypeptide, and entity number 2 is a bound ligand. We are not interested in the bound ligand. We are only interested in chaperonin. So programmatically, what we can do is we can check for molecule type and only proceed if the entity is a polypeptide entity. And if the entity is a polypeptide entity, we can store some useful entity information. For example, we can store entity ID here. and maybe molecule name as well, because that's always interesting. And most importantly, we want to look at how many copies there are. And the field that does that is called in chains. And that field is here. And once we do that exercise, for this particular entry 1kp8, we find that the entity that we want is entity number 1. The molecule name is 60-KDA chaperonin, which is correct because that is what we're interested in. And if we look here, it says number of copies is 14, and the chains are these chains, 14 chains. So now we have obtained all the chains of this protein within this entry ID. Now let us make the quality profile for all these 14 chains. And to do that, we need to take a look at the validation tab. And the endpoint that we are interested in is called RAMA sidechain listing. So go to the same page, but now move to validation tab. Again, you can input this PDBe ID 1kp8 into the validation tab and simply copy the URL. This is the output. Let us go through the exercise of going through this JSON output now. So let us refresh what we want to do. What we want to do is build a Ramachandran and sidechain profile. First of all, we need to store some data because this is in the form of quite nested JSON. So first of all, if entity ID is not 1, because we know we are only interested in entity ID number 1, we simply continue. Otherwise, we first take a look at the outer layer of the JSON output, which is the chain info. And then we take a look at the models, which is here. Within the chain info, we look at the models. And we are only interested in the first model here, because these are the same copy of a particular protein. After that, we look at the residue info, which is this part here, which is the meat of our data. What we can do for that is, because we want to look at residue-wise outliers, so from this level, we can collate 3 different variables and make it into a set of 1 variable. The reason for this is because residue can be represented by 1 number, which is residue number. However, if you use author residue number, then you have to take into account author insertion code. And that is what we are doing here. We are taking into account author residue number and author residue code, and putting in residue name as well, and making it into 1 variable in the form of a set. After that, what we're interested in is to relate the chain information. You cannot see the chain information in this page, but there is actually a field called chain ID if you were to scroll down. Let us store the chain IDs in 2 separate in 1 variable called outliers, but which contains 2 separate keys, one called RAMA and one called ROTA, the RAMA for backbone and the ROTA for the sidechain. And based on what is contained within the fields ROTA and RAMA, we can populate these variables, outliers RAMA and outliers ROTA. And the value for those outliers will be the chain information. So now we will know which chain has Ramachandran validation and which chain has sidechain validation. And it seems like all chains contain both validation, and that seems to be for all residues. Now we will build our profile. This is actually the same call as earlier, but a bit truncated. What we want to do here is to take a look at out of those 14 chains that we were looking at, how many Ramachandran states and how many sidechain states there are. If there are only one state for all of the chains, we're not interested in that because that seems to indicate a very normal validation. And if the length is, let us set a minimum value of 12 chains. We want to look at if 12 chains contain one state, whereas the other 2 chains contain different state than the other 12 chains, then that's what we want to look at. If this is the case, let us take a look at the popular state, which is what is inside the 12 chains, and the odd states. This is what you will get if you do this exercise. For example, this residue here, proline 462, 12 chains has the state called favored, whereas another 2 chains has the state called allowed. So these are actually the same protein, so we have to ask ourselves here, if we were to study this, this protein, why are these 2 not also favored? Does that mean that this is worthy of looking into? Probably. So based on this exercise, we can find out residues that need to be reviewed in the structure. And for example, here, alanine 3A3 are almost always an outlier, 13 out of the 14. Only in one model it is allowed. Does that mean that it is an actual outlier, or are these actually errors? So by building the profile, we can start to make educated guesses about the validation of our structure. We can do the same exercise for the side chains as well. So that is the end of my webinar. To register for future webinars, again, I remind you to go to this link or visit the events page. Next week, you will be hearing from my colleague, Srinath Nair, and he will be talking about the PDBe Graph API. Thank you for your attention.